Welcome to the Single Cell and Spatial Omics core of School of Biomedical Sciences. This video is giving a brief introduction of our service options and workflow for single-cell RNA sequencing and spatial omics by this core. Our core is currently providing a variety of single-cell full-package services. For details, please check on the Single Cell and Spatial Omics core section of webpage of School of Biomedical Sciences. For users who are interested in our services, a service form must be submitted with a sample declaration, confirming they are free of biohazardous risk. Single-cell RNA sequencing is a new technology which enables to simultaneously profiling of gene expressions in single-cell resolution. Compared to conventional bulk RNA sequencing, single-cell RNA sequencing is capable of new analyses on a sample, such as cellular heterogeneity, lineage tracing, and stochastic gene expression. The single-cell and spatial omics core provides a streamlined, cell-to-sequencing library single-cell RNA sequencing service package. Here is the workflow of single-cell RNA sequencing. At first, the sample is subjected to quality control, making sure that the cells are in single-cell suspension and free of clumps. Next, cells are being loaded into a microfluidic chip, together with RT enzyme and gel beads, before the final loading of partitioning oil. After the loading is finished, the chip is ready to be put into the 10x controller, where the oil droplets containing the single cells are being generated. A range of 500 to 10,000 cells can be placed in a single run. After the running is finished, oil droplets are being transferred carefully into a PCR tube for an isothermal reverse transcription reaction. Subsequent sequencing library preparation steps are followed. The constructed sequencing library is subjected to quality control steps, such as sizing and quantity check. The resulting library is compatible to Illumina sequencing platforms, where more information can be found in the web page. Currently, we do provide and subsequent next-generation sequencing service on the libraries generated. Extra charges apply. As spatial omics is getting noticed and recognized worldwide recently, the core is adopting this methods of the year 2020. With a spatial omics solution, researchers may obtain another level of data based on the conventional single-cell RNA sequencing data. Currently, the core is providing two types of spatial omics solutions. The first one is the nanostring CUHK in situ sequencing, whereas the other one is the spatial transcriptomics from the 10x genomics platform. Both spatial omics solutions are utilizing different profiling mechanisms and producing different types of data, such as resolution, number of genes profiled, and types of tissues supported. The first part is the nanostring in situ sequencing workflow. Briefly, a complete CUHK in situ sequencing experiment contains 3 steps. The first step is pre-instrument sample preparation. The tissue will be bound with gene-specific probes and cell segmentation markers for later fluorescent readout. The second step is on-instrument cyclic readout for in situ imaging signals. Finally, the raw data is uploaded onto a specific cloud-based server for data analysis. Please bear in mind that this platform supports either spatial RNA or protein profiling on tissue sections. The second part is the 10x spatial transcriptomic solution, using the 10x Visium Sci-Assist platform. This assay utilizes probes targeting the whole transcriptome from either human or mouse, followed by capturing of RNA signal on capture areas with unique barcodes. In such way, each RNA transcript is barcoded with a spatial identity. Subsequent library preparation resembles the conventional single-cell RNA sequencing, where the final sequencing library is subjected to NGS for signal readout. Currently, there are 2 resolution formats, the standard non-single-cell option and the high-definition option with single-cell resolution. For both spatial omics solutions, the input tissue can be either in frozen section or FFP format, therefore increasing sample submission flexibility. There are some basic sample submission requirements to fulfill, such as sample processing method, section thickness, and location of section placement. Here is a brief summary of the two types of spatial omics solutions that our core is providing. The single-cell and spatial omics core is welcoming collaborations and discussions on research projects. If you have any questions about our core, please feel free to contact our staff in charge as shown here or on SBS website.